Welcome students. Today we are going to see about a very important chapter. Is a part of the chapter which we are the 5th semester student, about extraction, isolation and characterization, in which last class we have seen general definition for chromatography and the topic about the column chromatography in the examination point of view. Today also we are going to have a discussion about the thin layer chromatography, and also I will continue with paper chromatography also today in this chapter especially. And when the students performing the practicals, it will be more helpful and more ideas you will get so that it can be expressed in the examination. And you will have a practical experience is more important than the theoretical working. As this thin layer chromatography and paper chromatography are the basic chromatographic techniques which are more applicable for various research. Almost all our research, chemistry or natural chemistry or even any herbal pharmacological study, we need to know about this instrument and the application. It's a very simple instrument, can be performed if we have the basic knowledge. See, here I have taken the original picture of the equipment which is available in our lab. The picture even you can see directly also when you come to the lab. And you see here, there is a telescopic metal where we can feed the slurry, and it is a spreader which is used for spreading the thin layer over the glass plates. And even you can see this one is a mica which is kept with a glass plate in the top, so that when you put the slurry of silica gel or aluminium or whatever this adsorbent you want to make it thin layer in the glass plate can be supplied, can be spread by means of this instrument which I have shown, spreader. And the third one, the third arrow which shows after spreading, after drying the thin layer, it can be the trays holder actually. The tray is for glass plate or the thin layer plate holder, and this is the important glass chamber, bigger one like a trough. It is the developing chamber. It will be having a glass lid also, and also sometimes aluminium lid or a silver or a metal lid also will be there to close, to make it saturation with the volatile solvent or the mobile solvent. Saturation is needed for the developing chamber. And this is the sprayer, the glass sprayer with the pump, rubber tube, and the pump which can be manually, we can make it a spray, which is a sprayer for developing or no, after developing, for detecting the spots. We need various detecting fluid have to be sprayed, like diclofenac, for example, for alkaloids we will use to drag and drop reagents, and like ninhydrin, many other detecting solutions which can be sprayed. Excuse me. And the same picture again, to make it more visible, I have just made the spreader again, and you can see the spreader, and this is the glass plate, and that is the mica sheet where the glass plates will be kept, and so that we can use the spreader. And this is the trays holder, the thin layer plate holder, and this is the developing chamber, glass chamber, and this one is the what I want to say, that is a sprayer. So now we will go to the explanation for the thin layer chromatography. TLC, usually we used to say TLC commonly in a short form. It will be having a trough and also slide, usually the thin layer plate after spotting the sample we used to keep in 45 degrees so that the capillary rays of the solvent system or the mobile phase can move through the stationary phase of the thin layer in the developing chamber. That's the picture. TLC is an example of adsorption chromatography. This you have to note it down. In another slide I used to say it can be followed in other system also. So how? So first, usually we are using a adsorption chromatography by using a adsorbent like say gel or alumina. So it is an example, you can say adsorption chromatography. It can be changed for other principle also according to the changing the adsorbent or the thin layer. The stationary phase being a thin layer adsorbent held on the suitable backing. Backing means suitable backing with the help of a binder. It needs a calcium sulfate or a gypsum is needed. That's why silica gel G is usually used as an adsorbent for making thin layer in a thin layer chromatography. Separation of the compound present in the plant extract depends on the differences in their adsorptive or desorptive. Adsorption means it's a surface phenomenon. So according to its affinity, according to the capacity of adsorptive power and desorptive power behavior in respect of the stationary phase. What adsorbent you are using according to the affinity of the phytochemical which you are interested to separate in a mixture, according to its adsorptive power or desorptive power behavior, they will be get separated. So TLC involves a thin layer of adsorbent mixed with a binder such as calcium sulfate, which is spread on a glass plate allowed to dry. The plant mixture to be separated, or which is your objective to be the mixture of compound to be isolated, separated, is applied as a spot near the base of the plate. Very, very important. Base of the plate, but it should not touch the mobile solvent. That always you should be careful. So, base of the plate you are keeping a spot or a band which is then placed closed. Glass tank, that is a developing tank containing a layer of developing solvent, that is called eluent, or that is called the mobile solvent, which is kept a line which you can see in the vessel in the picture which I have shown there. So this is the chamber, original chamber, where the plate is there and you can see the solvent very clearly in the bottom. That is the mobile solvent or eluent. The solvent moves up the plate by capillary action, carrying with its less strongly adsorbed component of the mixture. The components of the mixture which you kept as a spot in this thin layer will have different adsorptive power that we told. So some of them are strongly adsorbed, some of them are weakly adsorbed, some of them are mediumly adsorbed. So that the strongly adsorbed will be slowly desorption takes place. Medium adsorbed substance will be mediumly desorption takes place. And weakly adsorbed substances will be very fastly desorption will takes place. So the component will be carried by the mobile phase against the gravitational force by the capillary action in the thin layer of the adsorbent which you kept in the glass plate. The movement takes place, while the more strongly adsorbed component remain near the base of the plate because its movement will be less, desorption will take time. When the solvent have reached 1 to 2 centimeter from the top of the plate, it should not go above the top of the plate, it should not go away from the top of the plate. Only after 1 to 2 centimeter you have to stop. You have to remove the plate. Remove the plate. The plate is removed from the tank and dried. The now separated components of the mixture appear as spot on the finished plate. Chromatogram. You can see that chromatogram. This is 0 time and this is after 30 minutes. Spots can be seen if it is a colored substance. Otherwise, you have to spray a reagent to detect or to identify the compound corresponding to the bands of the adsorbent column. TLC, you can see the plate which I have seen the picture. This is small microscopic slide also. We can make thin layer plates and we can do the beaker, glass beaker as our developing chamber. The mobile solvent can be taken in the beaker and you can perform the thin layer chromatography for separation of mixture of compounds, for separation of mixture of phytochemical constituents which is isolated from the extraction from the herb. So with adsorption TLC, different substances have different adsorptive capacities and any one material can vary in its activity according to the pre-treatment of the thin layer plate. The adsorbent must be chosen in relation to the properties of the solvent, the mixture to be separated, the phytochemical which you are going to separate. In general, if a highly active adsorbent is used, then a solvent with a corresponding high power of elution, high power of solubilization, high power of elution for this substance will be required. For example, aluminium acidic is available, neutral is available or basic with different activity grades in commonly used. To produce a film with reasonable handling property, the adsorbent may be mixed with 12% of weight of the calcium sulfate is the gypsum as a binder to act as a binder to make the thin film in the plate. You can see that the picture which I have taken is there is a spreading type normally by manually which we can use the slurry in the micro slide and you can spread it into a thin plate and dry it and make activation and ready for the thin layer chromatography. Ready mixed powder, the adsorbent, are commercially available. They need to be mixed with a certain amount of water and may be spread onto the plate with a glass rod or by spreading by moving the slide as shown in the picture. We can get a very thin slides with coated as I shown in the picture. Even aluminium foil coated ready-made slides are also available in the market. The film set within a few minutes and then activated by heating at a suitable temperature. What temperature? Silica gel absorbs some vapors and becomes inactive actually. So it must be activated that unwanted vapors are from the atmosphere should be removed. Air drying this TLC plates for a duration of 30 minutes first. Then put it in the oven maintaining at 105 degrees centigrade for 30 minutes and then cool them in a desiccator, not in the open air, so that you can make the slide more active and to get separate the mixture of compound very easily. Solutions to be examined or applied to the film with the help of capillary tube or a micro syringe and micro pipettes or micrometer pipettes with measurements are also available. The amount which you have kept the spot can also be have determination. TLC, the solvent which we have discussed about, that you should know about almost all the solvents, and its polarity is needed, and its polarity is very much needed for you to know to study. Solvent used in TLC must be pure, analytical grade. Commonly used solvents are methanol, ethanol and other alcohols, chloroform, ether, ethyl acetate and many other, hexane, benzene, chloroform and carbon tetrachloride. These all according to the from low polar to high polar solvent system. You can gradient system can also be followed, or isocratic system also can be followed. According to the column chromatography, we have discussed about the solvent system. Visibility, detection of the spot of the compound. How? If invisible, the spots may be made into visible by heating for a specific period, examining under UV light if the substance are fluorescent. UV light examining, this is a UV chamber, interesting chamber which we can observe. The plates can be kept after the development. You can keep it inside and you can observe the spots very interestingly, like this picture which I have shown. In natural light, you can see the spot, the A picture. And if the UV light, if you put the yellow color switch, it will give the light of UV light in 254 nanometer. You can see this fluorescent spots. And you can even by another switch, the red color, you can find out the UV light in 366 nanometer, so that you can get the spots in the C picture which I have shown there. So you see the spots very clearly, you can observe. Even this is also visibility of the spots, like A is natural light, B is by UV light in 254 nanometer and C is by UV light in 366 nanometer. So the spots can be easily made visibility. Even by spraying the finished chromatogram with a suitable reagent, example of iodine, drag and draft reagent are used to spray, and many other reagents. Iodine can also, specifically all alkaloids will give you a brown yellowish spots. We can have an identification. Apart from that, you can go for many other reagents can be sprayed. The first one which I am showing is for iodine chamber. A glass bottle with iodine bits, vapors can be developed inside the chamber, and the spot, you can see the A, B, C, D. It's come to the D, keeping the slide inside, and you can see the brown, yellowish brown spots of alkaloid separated in the TLC plate can be seen. And sprayer also can be used. This is a sprayer. The initial slide which I told, this is sprayer where drag and draft reagent or ninhydrin, if it is an amino acid separation, or anisole you can use for any other alkaloidal compounds or glycosidal compounds. Specific detecting reagents can be sprayed by means of this sprayer, and so that you will get the spots in the different color according to the specification given by the qualitative chemicals which you have used. Advantages of TLC over paper chromatography. You should know that. What is it advantage? Thin layer chromatography is a glass plate with a thin layer of adsorbent, which is used for separating of phytochemical constituents from the extracted substance in a mixture can be separated. Separation of compounds can be achieved more rapidly with less plant material in TLC. The separated spots are more compact and clear, demarcated from one another. Reagents such as concentrated sulfuric acid would destroy the paper chromatography, but we can use in TLC, we may be used in the locating and separating the substance on TLC plate. And even simple and expensive when compared to other experiments, even paper chromatography also. Quick result can be achieved within between 30 minutes to a few hours. Good separation of spots and compounds have been identified. Very sensitive. The chromatography is resistant to action of chemicals used for the visualization of compounds. Components of the TLC now, after giving that explanation, we can make it into three components, which is major. The adsorbent stationary phase, eluent, that is the mobile phase of the developing solvent, you can say, and the third one is substance required to be separation, you are keeping the spot, that is sample, plant sample. And the fourth one is also there, developing sprayers and UV light which are needed for you to identify the spot. Also we need to find out the fourth component, but the main component 3, adsorbent is needed, eluent is needed, and also the plant spot, you have to need the sample which you are interested to separate, you need it. First, adsorbent. The adsorptive capacity of the adsorbent are related to their polarities. The more polar the adsorbent material is, the better it will separate compounds. Example, aluminium and silica gel. Under normal conditions, these adsorbents hold little water. Adsorbed organic materials, these decrease the adsorptive capacity of the materials. So they need activation. Heating may activate the adsorbents, and we have given the specification just 2-3 slides before. And the eluent, when you talk about the eluent, is the mobile solvent which is running through the stationary phase as shown in the picture. The more polar the solvent, the better is eluting, desorption property. Therefore, water is the better eluent than ether. Ether which is better than the cyclohexane. Why? Because cyclohexane is least polar, better than cyclohexane. Next high polar is ether, and the next high polar is water, which we have given the 3 examples. So that's what they want to say, the power of the eluting power. The substance required separation, polarity also plays an important role. The least polar substance, especially saturated hydrocarbons, are least strongly adsorbed, while the high polar compounds like acids are strongly adsorbed. So this is what we should have an idea. In practice, only 2 adsorbents are widely used, that is silica gel. In thin layer we used to see silica gel G. Aluminum. When used these adsorbents, the rule is to match the polarity of the developing solvent with that of the compounds of the mixture to be chromatographed. RF value is very important factor which you are here. RF value means distance traveled by the substance or the sample from reference line, that means the baseline in centimeter, divided by distance traveled by the solvent friend from reference line. Usually we used to write distance traveled by the solvent friend divided by the distance minus we want to find out the distance travel, how much the sample travel divided by the distance traveled by the reference line. That gives you always less than 1 usually, RF value. So RF value is very, very important for getting the samples identification by TLC. Why do you need RF value? A question. The chromatography RF value are the most basic prerequisite of the experiment. These numbers indicate whether the analyte or the solute or the sample prefers the stationary or the mobile phase, the solubility with the mobile phase. With the stationary and the mobile phases, RF value are used to determine polarity, relative masses and relative solubilities, among other things. So this is what the basic thing the RF value will give you. Separation of alkaloids, an example. Alkaloids need a moderately polar solvent for good separation. So this is a common, there is different types of solvent system is used, different type of solvent system. But example, ether and ethanol in the ratio of 95:5 is commonly can be used for some alkaloids. So you will get a very good spot separation of the mixture of compound if you use a mobile solvent in the ratio of ether and ethanol. In adsorbent, I did not say silica gel G can be taken. A right decision. But in case you should know about the problems also, no? More polar solvent, suppose you are using completely methanol. Instead of ether and ethanol, you are using completely methanol. Would be preferably absorbed. The alkaloid should be carried along the passage of the solvent. They may not get separated properly because of its high polarity with the alkaloids which you are interested to separate, resulting in a poor separation as shown in the picture. You see the spot? Not getting the proper separation because we used methanol as a mobile solvent. Even it can be like this also. So another one possibility. On the other hand, if a person is using non-polar solvent like cyclohexane, that is methanol, you are getting this type of wrong, and you are using, okay, I will use very low polar solvent, cyclohexane, would be unable to display, it will not unable to displace the alkaloid. It cannot move also from the baseline. And the separation may not occur. From the adsorbent layer, they would then remain at or near the baseline or the origin itself as shown in the picture. So this is the second choice. This is also possible. So you should have to use a mobile solvent or the eluent in the proper ratio for every phytochemical compounds. This is a simple example I have told. Additional factors for separation of alkaloids. If using an aluminium thin layer, neutral, a neutral solvent should be used. If using silica gel, slightly acidic due to the method of preparation, and alkaline solvents such as acetone, water, 25% ammonia in the ratio of 90:7:3 makes a good separation. This is also possible for you to do. Separation of volatile oil, an example. The constituents of volatile oil are mainly nonpolar terpenes and are therefore best separated with corresponding nonpolar solvents such as chloroform benzene mixture. Certain oils may be more polar solvents, clove, oil, phenolic. They also can be done by system. Separation of sugars and sugar acid mixtures can also be done as shown in the picture, the separation. This mixture produces by hydrolyzing starch and gums. They are strongly polar and are therefore strongly adsorbed on the silica and alumina layers. Although strongly polar solvents are used, separation on these thin layers is not generally satisfactory, and better results are obtained using weakly polar adsorbents such as cellulose and a polar solvents such as butanol, ethanol, water in the ratio of 5:4:1. Alternatively, paper chromatography can be used. But usually TLC is the best one. TLC is the comprehensively better separation tool than paper chromatography. Though the adsorbent term use various types of TLC, but principle of separation may be any one of four fundamental mechanism of separation is possible. Even though we say we are using an adsorbent of silica gel, alumina, and so that it works in adsorbent chromatography, but you can use ion exchange chromatography in TLC also by changing the adsorbent as an ion exchange resin. Partition can also be followed by means of coating with the cellulose molecules which contain water molecule incorporated or embedded with the molecules can also be used as an partition chromatography. Adsorption chromatography, as we know, alumina or silica gel G, whichever we are using as an adsorbent, as a thin layer, that can also be followed. Molecular exclusion by means of dextrin coating with a gel, it may work as a molecular sieve, and molecular exclusion chromatography can also be followed in thin layer chromatography. Excuse me. Principle of separation totally depend upon the coating material or coating substance applied on the plate. This is an important point for the students to remember. Principle of TLC, principle of any chromatography depends upon the coating substance applied on the plate. Stationary phase and the mobile phase decides the principle of separation of a chromatography. If silica or alumina is applied as coating substance, adsorption principle will take place. If coating substance is Sephardex or dextrin, molecular exclusion will the principle. It acts as a molecular sieve of separation. If your resin, ion exchange will be mechanism of separation if resin will be the coating substance. And if cellulose like coating substance, then automatically it will adopt partition mechanism. So you can have all the four types of mechanism of chromatography in thin layer chromatography, how you are changing the coating material. So this explanation you should write. Suppose if they are asking a short notes, write about TLC, the basic principle behind the thin layer chromatography, or basic principle behind paper chromatography, basic principle by HPLC. So you should write very clearly about the stationary phase, mobile phase, and how the separation takes place. What is the principle behind? And this is the spreader which I want to show you, because we are going to find out what all the different types of, other than this spreader, pouring type, spraying type is also available. The most often adsorbent are applied on the glass plate. The dimension of the glass plate which is used for TLC may be vary from 20 into 20 centimeter or 20 to 5 also, lengthy glass plates. Sometimes even microscopic plates which you are using as shown in the picture of the previous slides are also used to examine the process of chemical reaction by thin layer chromatography method. The glasses are coated with adsorbents with different techniques like pouring and spreading, dipping the slides into the slurry, spraying the slurry glass plate, and spreading technique as shown in the picture. The spreader can be used or a glass rod also can be used. Also then air dried and activated them in 110 to 120 degree for an hour they have given, but usually 105 for 30 minutes will activate them after air drying. Sample can be applied by micropipette or capillary tube. Capillary tubes, which you know with even measurement, is also available. This is micropipettes with measurements, with bands of pipettes are also available with samples. 4-5 samples can be dropped at a time. It should be spotted at least 2 cm above the base, because the mobile solvent or the eluent should not touch the sample spots, or on such height that spot area should not immerse into the mobile phase. The main concept is it should not immerse in the mobile phase. You can see the spot that is called baseline. Our teacher used to find out for make us to first experiment to draw the baseline, even in the paper chromatography or even in the thin layer chromatography also, and the development, and then you will get the spot if it is a colored one. Otherwise you should go for detecting reagent spray. As in this picture you can see that plate will be kept in a 45 degree angle. The spots are kept in the baseline and you are getting the spot inside the chamber after developing. The area of the spotted sample should be minimum with sufficient concentration. Mobile phase and TLC, thin layer chromatography, should be kept in development chamber. The chamber should be saturated. Why the word saturation? Very important with mobile phase. Otherwise edging effect or edge effect will take place. What is edge effect? This is the edge effect. The solvent, after moving to the front, you see that curve like a bump. So this is due to the unsaturation of the mobile phase in the developing chamber. This is a problem. The height of the solvent cannot be seen properly, because RF value, when you want to calculate distance traveled by the sample divided by the distance traveled by the solvent or the mobile phase. So the chamber should be saturated with the mobile phase. Otherwise edge effect may found, in which the solvent front in the middle of plate moves faster than the edge. The mobile phase selected on the basis of phytochemical constituents which you are interested to separate, which have to be separated, and the nature of the stationary phase. It may be single or the mixture of solvent. The polarity of the solvent adjusted such that it easily separated the phytoconstituents. So it's very important to control this. How can you control this edge effect? How can you prevent this edge effect for your experiment? Edge effect can be prevented by saturating the developing chamber. So this is a chamber, you can saturate it. The mobile solvent can be kept, and the sides you can see the B, picture B. You see there sides a blue color line. That is filter paper which is already dipped with the mobile solvent, and the lid will be closed for some time, so that the mobile solvent will be supported, the vapors will be, there will be an arrow which is shown there, so that this edging effect, this curve will not appear. The normal phase, how you are getting a straight line of the solvent front, while it is running from the base to the top by means of capillary action, you will get a good result. Even the separation of the compound will be the correct one. RF value will be, uniformity will be there. The chamber with normal saturation, the arrow stands the vaporization of the eluent from the layer, and the dots symbolize the vapor density, which I have shown there in the picture. The chamber lined the sides, the filter paper saturated with eluent vapor, so that you will get a very good saturation in the chamber, so that the edge effect can be prevented. The different development techniques used in TLC are one-dimensional, two-dimensional, horizontal and multiple development techniques. So in one-dimensional and or vertical technique, the mobile phase flows against the gravity. That is the normal one which you are due to the capillary action. Most separation are done by this technique. It is one spot is applied at the corner of the plate, and the plate are developed in one axis, then dried it. And but in the second one, further, this is the second one, the picture, one direction and another direction. So the plate will be 90 degrees again developed. Further turn the plate for the next another axis, so that it become two-dimensional plate. And you can see one-dimensional, you are getting the spot in this manner, and when you turn it, this is the first direction, the spot, and this is the second direction. So the individual spot again it is getting separated. So more constituents mixture can be separated by means of this two-dimensional technique. After development of the chromatogram, the isolated compound can be visualized by detecting agents that we have discussed already. The detection agents can be of two types. One is destructive type, irreversible. Non-destructive type is also there. Recovery. In destructive type, the sample cannot be recovered, or it will be destroyed due to the chemical reaction of the spray reagent which you have sprayed on the sample. While in the non-destructive method, the sample can be recovered. And non-destructive method, samples can be even detected by UV chamber method, densitometric method, or iodine chamber method. UV chamber I have discussed about that, and iodine chamber also I have discussed. Densitometer means this one, directly you can digital. What is the density of the color, what is the density of the amount present can also be detected. Very quantitative estimation. For quantitative purpose, the density of the spot can be measured, or the spot can be eluted even with a solvent and analyzed by conventional technique like spectroscopic method or spectrophotometric method, and electrochemical methods can be used. Now we are coming to the next one, paper chromatography, which is closely related to the thin layer chromatography. Here already the paper is taken as the thin layer. Paper is hanging, the solvent is present as in the thin layer, and solvent friend will be run as we have decided up to the 2 cm below to the top. The cotton thread, it will be hanged as a clipped, and solvent friend, filter paper, solvent tank which we are telling that is the developing tank, spotting line, solvent system, and the direction of the solvent flow, that is ascending paper technique. Even descending is also there in paper chromatography. Paper chromatography is carried out by specially designed filter paper. In general, paper chromatography refers to partition principle. In general, it is refers to partition principle. How? Very simple, the students should listen now. Paper is made up of cellulose. Cellulose molecules contain water surrounded by each molecules which cannot be visible normal eye. So the water molecules is a stationary phase, and another phase, mobile phase, is also a liquid. So liquid means automatically you know that is distribution of the sample between the two solute, between the two immiscible liquids, they are called partition. So the technique is followed by partition. You should not have any confusion. So the paper is solid. So how come it is partition? Paper is made up of cellulose. Cellulose molecules are surrounded by water. You are taking a mobile solvent, it's a non-polar. Polar is stationary phase and non-polar is a mobile phase which you have taken as a eluent. So that it will work in partition chromatography. Paper is always partition principle. Unless otherwise, if you are coating with the paper another adsorbent. The principle of separation may be here partition or adsorption. Another question, or adsorption. If the filter paper is impregnated with alumina, suppose you are spreading the paper with alumina or silica as an adsorbent principle, if it is coated, the paper is coated, then it will work in adsorption chromatography. Otherwise, simple paper, cellulose, it will work in partition chromatography. Will apply the separation, whereas the moisture of water present in the pores of the cellulose fiber, it works as a stationary phase and the solvent as mobile phase, then the principle of separation will be partition. So we know very well, last class itself we have discussed about the basic chromatography. In that, partition means distribution of a solute between two immiscible liquids. So two liquids are there in a chromatography application, then it will work in partition chromatography. So this is the paper with colored spots, and a beaker have been used as a developing chamber. Various types of filter paper are used for paper chromatography. It may be Wattman filter paper. Different grades are there. Acid or base wash filter paper. Paper modified with glycol, formamide, methanol, glass fiber type papers are available, and hydrophobic paper with OH group can be acetylated, or paper can be impregnated with alumina, silica gel, and also even impregnated or coated with ion exchange resin. So the size of the paper should be suitable for the size of the chamber which you selected. Apply the sample with a capillary tube or micropipette. The sample should be dissolved in a mobile phase and apply with low concentration with small zone. Usually we can perform this in our lab for mixture of amino acids. Use an anhydrin as a developing or a detecting sprayer, and a solvent system, N-butanol, water, acetone can be used as a solvent system for mobile phase. You can find very good result which we can perform in the lab. In the mobile phase, pure solvent of mixture of solvent or a buffer solution can be used. There may be ascending, descending, circular, two-dimensional and ascending, descending type. Together, different techniques are developed now. Ascending development technique is conventional, normally used technique, in which the mobile solvent moves against the gravity as you did in TLC, and the spot of the sample will be kept in the bottom portion. While in descending development, the mobile solvent will be kept in a trough in the top, and the flow of the liquid from the top to bottom. Here, the paper will be hanged. Here, the sample applied in the top of the developing area in fast due to the gravity will assist the solvent flow. As shown in the picture, lid will be closed. Trough will be having the mobile solvent. Paper will be hanged from the trough to the bottom. Spot will be kept in the top, and so that you can see mobile phase runs down and you will get very faster separation. In circular or radiant development of sample applied in the center, the paper can be mobile phase flown through the wick, that means a wick like thing which will be in a circular petri dish, at the center and spread uniformly in all directions as shown in the picture. You see the red color wick with a paper which will be kept upside down in a petri dish as shown in the picture, so that the wick will suck the mobile solvent and then circular movement can be seen. The sample spot will be kept in the center. Circular filter paper will be having a separation. The RF value can be calculated as a diameter can be done. The two-dimensional development techniques, and the sample is applied in one corner and the development of the paper in one axis and dried the paper, as we have seen in the TLC also. After drying the paper in 90 degrees angle, development the paper in the another axis. So the technique can be used for more complicated compounds which we want more separation, then we can go for two-dimensional. The ascending descending development, both will be there. Separation can increase actually. One will be ascending, another will be descending. First will be ascending takes place and descending development as shown in the picture. Ascending descending. The paper will be kept in a triangle, both the trough. See, from here the solvent is going down. So that is also one type of technique, ascending and descending technique. After development of the chromatogram, the isolated compound can be visualized by detecting agents. Detecting agents can be are of two types, as we have discussed in the TLC also, destructive type and non-destructive type. We know very well. In destructive type, the sample cannot be recovered. It will destroy due to the chemical reaction which is sprayed the reagent with the sample, while non-destructive method samples can be recovered. In non-destructive method, samples can be done by UV chamber, densitometer method or iodine chamber method, which we have discussed even in the in the thin layer chromatography. Paper chromatography can be used both quantitative and even qualitative purposes. The qualitative purpose of RF value can be determined. RF value factor, you know that distance traveled by the solute or the sample divided by distance traveled by the solvent friend. So that is, when you do the perform in the lab, you have to write the experiment yourself. I will come there personally and I will make you write the procedure what you are performing for paper chromatography and thin layer chromatography. That will make the students to completely understand than this theoretical study. Practical will give you more advantage for you to understand and to use in your future career. Thank you. Next class we will see about the next chromatographic technique. Thank you.